Thank you very much, Captain Stoddard. And thank you to everyone in CDER's incredible Small Business and Industry Assistance Group for yet another remarkable event. Workshops like this facilitate collaboration between FDA and generic industry stakeholders, and this annual Science and Research Public Workshop fulfills one of our commitments under the Generic Drug User Fee Amendments GDUFA. Collaboration allows us to prioritize scientific challenges and knowledge gaps that once addressed by research support activities that facilitate generic drug development. GDUFA Science and Research is the beginning of a dialogue that can eventually lead to a generic drug approval. The science and research described in today's workshop are the foundation of an integrated system that culminates in the availability of generic complex products. Results from the research projects that you help us plan today need to be translated into answers to the questions that are most important to the generic drug development industry. And we have many more events coming. Recently, FDA funded a Center for Research on Complex Generics, the CRCG, which is a joint endeavor that includes faculty from the University of Maryland, Baltimore, and the University of Michigan. The CRCG has engaged directly with numerous generic drug industry stakeholders to glean detailed insights into the most pressing current scientific challenges impacting generic drugs. We are excited to have the co-directors of the CRCG here with us today to share their insights with us in a presentation this morning. The CRCG will facilitate communication and information sharing among FDA, academia, generic drug developers, and other generic industry stakeholders. Some of this will be in the form of virtual events and workshops. I hope you will join us at our upcoming virtual events, like the in vitro release test and in vitro permeation test methods best practices and scientific considerations for ANDA submissions, which will take place in August, and Advancing Generic Drug Development Workshop, Translating Science to Approvals in September. And now, I'm honored to present our keynote speaker, Acting Commissioner Dr. Janet Woodcock. Dr. Woodcock began her long and distinguished FDA career in 1986 with the agency's Center for Biologics Evaluation and Research as Director of the Division of Biological Investigational New Drugs. She also served as CBER's Acting Deputy Director and later as Director of the Office of Therapeutics Research and Review. In 1994, Dr. Woodcock was named Director of the FDA's Center for Drug Evaluation Research, CDER, overseeing the center's work that is the world's gold standard for drug approval and safety. In that position, she led many of the FDA's groundbreaking drug initiatives. She has also served in other leadership roles at the FDA, including as Deputy Commissioner and Chief Medical Officer. With the onset of the COVID-19 public health emergency last year, Dr. Woodcock was asked to lend her expertise to Operation Warp Speed, the initiative to develop therapeutics in response to the pandemic. Dr. Woodcock was named Acting Commissioner of Food and Drugs on January 20, 2021. She also has received numerous honors during her distinguished public health career, including a Lifetime Achievement Award in 2015 from the Institute for Safe Medication Practices, the Ellen V. Siegel Advocacy Leadership Award in 2016 from Friends of Cancer Research, the Florence Kelly Consumer Leadership Award in 2017 from the National Consumers League, the 2019 Biotechnology Heritage Award from the Biotechnology Innovation Organization and Science History Institute, and the 2020 Lifetime Achievement Award from National Organization for Rare Disorders, NORD. And I also have to mention that she's an avid and accomplished gardener. Thank you for joining us today, Dr. Woodcock.
I'm pleased to have this opportunity to address the 2021 Generic Drug Science and Research Initiatives Public Workshop. I'm sorry we can't be together in person, and I look forward to that day soon. For well over a year now, the COVID-19 pandemic has dominated our personal and professional lives. Across the FDA, I've seen the strength and resilience of FDA staff who quickly applied their experience, preparedness, and expertise to implement new systems and processes to respond to the COVID-19 public health emergency, while continuing to meet our many regular and critical responsibilities. And the generic drug program was no exception. For example, our Office of Generic Drugs addressed the potential impact that the public health emergency might have on abbreviated new drug applications, bioequivalence studies, and the submission of ANDAs to FDA for assessment. Our Office of Pharmaceutical Quality worked with manufacturers of approved generics who needed to make changes to manufacturing processes and facilities that experienced disruptions from the pandemic. And FDA prioritized the assessments of generic drug submissions involving potential treatments and supportive therapies for patients with COVID-19, even as we continue to assess other generic drug applications. FDA has already approved more than 900 original and supplemental ANDAs for these critical drugs. All the while, FDA's Science and Research Program, established under the Generic Drug User Fee Amendments, or GDUFA, has continued its important work. Good science and rigorous research are the lodestar of the FDA, and they're a critical piece of our work to support and modernize the generic drug program and the generic drug industry, and to help ensure the continued supply of safe and effective generic drugs to the American public. Now, we know the importance of generic drugs to the public. Patients today in our country fill 90% of prescriptions with generics, and many of these are critically needed, often life-saving drugs. Generics also mean enormous economic savings. In the past decade, generic forms of drugs have saved more than $2.2 trillion in public health costs. The Generic Drug User Fee Amendments, GDUFA 1 and GDUFA 2, have provided direct and essential support for generic-related science and research at the FDA, and help ensure that we continue to improve the efficiency of generic drug development, assessment, and approval processes. During previous authorizations and reauthorizations of this law, we've worked productively with industry to strengthen it. We continue that tradition now as we work toward the latest iteration. GDUFA-funded research was intended to accelerate scientific and technical advances for generics, to modernize evidence-based bioequivalence standards, and to ensure that regulatory concepts are compatible with the innovations in drug development that have occurred across the last several decades. Quite simply, the scientific research funded by GDUFA helps build an essential bridge from the scientific knowledge of the 20th century to our work as an effective health and consumer protection agency for the 21st century. To do this, however, we need to continue to expand our scientific and data capabilities. That's why, for instance, the FDA recently began an important effort to become even better informed, more efficient, and better prepared by launching the Technology Modernization Action Plan and the Data Modernization Action Plan. It's really essential that FDA's regulatory decision-making is based upon the most current scientific evidence, that bioequivalence standards are as efficient as possible, and that the approaches to develop generic drugs and assess ANDAs incorporate 21st century technologies. Nowhere is this need more obvious than the development of complex generic drug products. The Generic Drug Science and Research Program has delivered new approaches in many categories of complex products and has allowed the FDA to build a scientific and evidence-based foundation for the feasible and efficient development of these products. Generics of complex brand name drugs or reference listed drugs can be more difficult to develop, such as those for peptide drug products, dry powder inhalers, or topical medicines, to name a few. The historical difficulty to develop complex generics, compounded by the inefficiencies that have limited generic competition, means that a complex drug product is less likely to have an available generic. Insights gained from GDUFA-funded scientific research have enabled the agency to articulate viable and scientifically sound alternative approaches for industry to demonstrate bioequivalence for these complex generics. Consider a few recent examples. In December 2020, the FDA approved the first complex generic of glucagon to treat severe hypoglycemia, i.e. very low blood sugar, in patients with diabetes. The brand name product was approved over 20 years ago, and this approval of a synthetic peptide generic product was finally made possible because of FDA's research on analytical methods for peptides and immunogenicity testing for peptides. In January 2021, FDA approved the first complex generic for a parenteral, i.e. not oral, iron product that treats iron deficiency anemia. The FDA's scientific investment in characterization and advanced bioequivalence study designs was essential to this approval. And in February 2021, the FDA approved a complex generic for loteprednol, etobotinate, ophthalmic suspension to treat eye inflammation using a new in vitro bioequivalence approach. Without the new approach, applicants would have had to recruit hundreds of cataract surgery patients to demonstrate bioequivalence. Investments in particle size characterization and eye models supported this more efficient bioequivalence method. As you hear about some of the research projects described in today's workshop, I urge you to contemplate what it takes for them to be translated into tangible and practical answers that support generic drug development. One early and important activity is productive communication between FDA staff and drug developers, including providing responses to controlled correspondence, issuing product-specific guidance, and conducting pre-ANDA meetings. These kind of interactions with prospective generic drug developers during product development help clarify regulatory expectations and help reduce industry's uncertainty about accepting and adopting new bioequivalence approaches. Our staff is able to efficiently carry out these essential activities because of the work done in the GDUFA Science and Research Program. During the ANDA assessment process, for example, FDA staff utilized the insights gained from GDUFA-funded scientific research to support regulatory decision-making. A central purpose of today's workshop is to facilitate the critical collaboration between the FDA and generic industry stakeholders. We can then appropriately prioritize specific scientific challenges and knowledge gaps that, when addressed by research, can support numerous activities that facilitate generic drug development. Both industry and the FDA benefit from the GDUFA science and research programs. Ultimately, however, it's the American patient who profits most, since these activities contribute to timely approvals of critical and cost-saving generic products. Continued robust investment in generic drug science and research will help modernize and maximize the use of the generic pathway to provide safe and effective generic drugs to the public. This research allows FDA to adapt and optimize our approaches and regulatory decision-making to ensure the availability of effective generic drug pathways, particularly for complex products. When drug developers use this pathway, patients are able to benefit from a proven system of high quality and more affordable substitution of generics at the pharmacy. Several presentations today focus on modernized approaches that use computational models, machine learning, and other state-of-the-art simulations that minimize the need for testing of generic drug products in humans. These are very interesting new ways of doing things. Numerous additional scientific challenges, of course, are on the horizon, but I'm confident that FDA's generic drug program will rise to the challenge. Today's workshop represents an important opportunity for engagement and collaboration to identify and prioritize how and where we invest our resources to prepare for a thriving and sustainable generic drug program in the 21st century. Thank you again for your participation, and I hope you have a very productive day.
Thank you so very much, Dr. Woodcock, for that inspiring keynote message. And it is now my pleasure to welcome Dr. Robert Lionberger, who himself will provide the formal introduction to the workshop. Dr. Lionberger is the Director of the Office of Research and Standards, ORS, within the Office of Generic Drugs, OGD, and in this role, he leads OGD's implementation of GDUFA regulatory science commitments, including internal research activities and external research grants and collaborations to ensure the therapeutic equivalence of generic drugs. In his 10 years as a member of the office's science staff, Dr. Lionberger's accomplishments include the development of bioequivalence methods for complex and locally acting drugs, mathematical modeling of drug dissolution and absorption, and incorporation of pharmaceutical development information into the ANDA review process. Please let us welcome Dr. Robert Lionberger.
Good morning. I'm Robert Lionberger, the Director of the Office of Research and Standards in the Office of Generic Drugs. And it's my pleasure to introduce today's workshop. We had great opening remarks from our OGD Office Director, Sally Choe, and our Acting Commissioner, Janet Woodcock. We thank them for taking the time to be with us here today. Science and research is an important part of the GDUFA agreements between FDA and the generic industry. It makes the entire generic drug program work better. Research helps FDA develop product-specific guidances for complex products. It ensures FDA staff are prepared for in-depth scientific discussions at pre-ANDA meetings. It pushes the development of characterization methods and modeling tools towards solutions that are relevant to generic drug development and can support alternative BE approaches. For complex products, the research is targeted to ensure that there's a path toward generic competition for every drug product. For all FDA products, research is designed to accelerate product development by industry and to make our ANDA assessments more efficient. This benefits the public through faster access to generic versions of all products. Today's meeting is important for achieving these goals because input from stakeholders and the public is very helpful to FDA as we identify the specific science and research activities that we will conduct. Input from the public and public health stakeholders helps identify areas where increased access to generic drugs will have the most impact. Impact from the generic industry and related pharmaceutical scientists helps us understand the scientific challenges and their potential solutions. There are 3 ways to provide this input. First, we will capture what our speakers, panelists, and attendees say at this meeting. Further written comments can be sent to the docket. This is also where you can follow up if this meeting leads you to develop some new thoughts. The Federal Register notice also describes the confidential comment process. We have designed this meeting to help this process and help draw out thoughtful comments from the participants and attendees. First, the background document on recently approved new drug applications can remind you of recent approvals and set an appropriate context. Our morning speakers from the Center for Complex Generics will provide an overview of their interactions with generic industry stakeholders. They've done a tremendous amount of outreach to the generic industry to help facilitate input. In the morning, we'll also introduce our breakout sessions with industry and FDA speakers for each session. After our brief lunch break, we'll have a plenary panel where we'll discuss some of the morning comments at a higher level. The afternoon breakout sessions will take deeper dives into 3 areas. Again, the goal of this meeting is to help FDA update its science and research priorities related to generic drugs. I have provided the link to our current priorities. Comments that help us refine and focus on specific issues within these priorities are always appreciated. To help frame your input, it can help to consider these two questions. Do these priorities provide generic pathways for the newly approved complex products? And do they help optimize product development and patient access for non-complex products? In our Federal Register notice for this workshop, we also asked 5 specific questions on which we would like input. We will give each of our panels and breakout sessions a chance to comment on these questions. Our first question touches on an area that's a little bit outside the current generic drug program, where injectable products with Q1 and Q2 differences from the RLD can sometimes lead to 505(b)(2) submissions rather than 505(j) submissions. We would like input on scientific questions relative to evaluating whether these differences impact the substitutability of products with these differences. Secondly, we've noted an increase in the recent NDA approvals that contain oligonucleotide drug products. For example, small interfering RNA, chemically modified oligonucleotides, antisense oligonucleotides. Like everybody knows that some of the COVID vaccines are also oligonucleotide mRNA-based products. Therefore, we welcome comments on this entire class as we attempt to identify any aspect of these products where science and research will help clarify the path to generic competition. Software may be eating the world, and we may be drowning in data. We'd like to hear what stakeholders think are the opportunities for artificial intelligence, including machine learning, and of the use of big data to facilitate and modernize the development of generic products. What research can FDA do that'll help use these tools in the generic drug development process? Next. We think that the GDUFA research program has been exceptional in its ability to bring new analytical technologies or modeling tools into the discussion around generic product development. From our internal use of these tools, they have allowed FDA to open many new in vitro-only BE paths. What we would specifically like input on during this meeting is things FDA can do to help speed the adoption of these new scientific tools into product submissions and eventually FDA approval of more complex generics. The Center for Complex Generics will have a key role in this translational aspect of the GDUFA Science and Research Program, but we'd like to hear your thoughts today on things we might do that will help integrate new technologies into ANDA submissions. Finally, we recognize that the generic drug industry is a global one. As ICH processes for generic drugs move forward, we'd like stakeholder input on areas where research and new data can help identify best bioequivalence practices and support convergence of global bioequivalence standards. We look forward to a world where there are more consistency in the bioequivalence approaches used in different regions across the world. We think this will make generic drug development more efficient and lead to, again, faster global access to high-quality generic products. Overall, I specifically look forward to your input and engagement with this workshop. I'll be hosting the noontime panel and having some direct live discussions with some key stakeholders. And our program will now continue with the leads for the Center for Complex Generics, Jim Polli from the University of Maryland, and Anna Schwendeman from the University of Michigan. Both are exceptional scientists and have collaborated with the GDUFA Science and Research Program in the past. I'm very grateful that they lead this center, and they'll be talking about the work the Center for Complex Generics has done to capture detailed input from a broad range of industry stakeholders, and look forward to hearing their presentation.
Thank you for that kind introduction. Again, my name is James Polli. I'm from the University of Maryland. And I'm really pleased to be here with you this morning to talk about results of a survey from earlier this year where many of you actually participated. And one thing I would just encourage you to do is to please visit the website of the Center for Research on Complex Generics. It's located here at the bottom of the screen, which again is a collaborative agreement between the University of Maryland, University of Michigan, and the Food and Drug Administration. One reason why I and Anna are so pleased to be here with you this morning is because we're really very interested in knowing what research you think is needed. And I know there's 5 critical questions for today. At some level, they might be able to be summarized as what research is needed, and which in part is what this survey is about. For example, this survey asked three questions in particular. What products are necessary for further investigation to help the development of complex generics? What techniques or tools are needed? And also what training is needed. And I think hopefully you'll find that the survey was very informative. But having said that, and Anna will talk about this more next with regard to some of the informational sessions that we conducted with companies. But in general, I'd say there's still a need for greater specificity in general, as far as at least us being able to help in terms of executing research and putting on educational programs and that sort of thing. Although obviously this center is really just less than 1 year old, though. Okay, so this is sort of an informational slide. So the mission of the Center for Research on Complex Generics is to increase access to safe and effective generic drugs through enhanced infrastructure and communication, education, and research collaboration across industry, academia, and the FDA. And this is maybe more of a slide for future reference in case anyone subsequently asks you, what does this new center do? And again, I would just encourage you to at least check out our website at Complex Generics, where we'll be putting more information up. One thing we'll mention is that we do have an educational program in August. That information, I think, is up by now, and more will be following. Okay, so one component is training. So, for example, we have plans to do webinars, workshops, hands-on laboratory demonstrations, student center events, public outreach, and complex generic scholars program. And I guess the one thing in this slide that would like to mention in particular is hands-on laboratory demonstrations. So, for example, at Maryland and Michigan, and certainly we look forward to other partners. There's lots of research and laboratory facilities, and we'd like to use those facilities to demonstrate critical techniques. But of course, the question is, which ones should we do? So just going back a couple of slides about how very interested in learning as much as possible today about, with some specificity, what research is needed, what training is needed. And again, for example, this survey was informative, but I would also say that obviously it doesn't answer all of our questions. Okay, in terms of what we do have planned, so this first year wasn't exactly the one that we had initially planned, but we are at the point where we've got some workshops planned, including coming up in August 18th through 20th. One in the topical dermatologic area, in vitro release testing, IVRT, and in vitro performance testing, IVPT methods, best practices, and scientific considerations for ANDA submissions. In September and October, one on mechanistic modeling, regulatory utility of mechanistic modeling to support alternative bioequivalence approaches. And then in late November, one is kind of more focused on long-acting injectable products, best practices for establishing the suitability of model integrated approach to demonstrate bioequivalence of long-acting injectable products. And I think at this point, all these are free and open to the public, and I think for this first one we do have at least preliminary information, including registration at complexgenerics.org. So please visit that website. Okay, and of course there's also a research mission. I think, for example, we do have capabilities, long capabilities at least the two universities to do industry-sponsored research. But of course, the center is a federally supported, FDA supported. So I can't say we do research that aims to help promote the interests of say, one company in confidence. So not here to really talk about that. Talk about things that we do sort of publicly. Again, I think I mentioned about laboratory demonstrations. So of course, there's some bleeding between research and training. Also interested in doing pilot lab projects, sort of research on a smaller scale about feasibility of methodology. I think the one I just want to emphasize a little bit is Research Scientist Program for OGD staff and generic companies. So again, kind of going back to the theme, what specifically do you think we can do to be helpful? If you think that there's, we certainly are interested in training the public, generic companies, including FDA staff. So if you think there's certain techniques that are maybe innovative techniques that it would be helpful for the OGD staff to be familiar with, we actually are very interested in that. And if you have specific examples of that, please let us know. Be glad to leverage the facilities that we have. In terms of research that's being just initiated now, there's two here. One deals with doing laboratory studies to better parameterize oral drug absorption mechanistic models. For like things like food effect, it's important how to incorporate micelle activity. So that's what that's about. And also another project with regard to better understanding of a liposomal injection product, including release testing. Okay, so that's a little bit of introduction about the center. Although maybe I forgot to mention maybe a critical part. Again, it's a collaborative agreement between two universities. And we're open to involving other experts at other institutions, the University of Maryland, University of Michigan, and the Food and Drug Administration, and which, again, the intent is really to add on to the portfolio of what OGD currently already does. So hopefully it adds value. Okay, so transitioning now to the main event here, talking about the survey of scientific challenges in the development of complex generics. We really had three main questions. Again, much thanks to many of you for participating in this survey. One question is, which complex products should we focus on? Again, we're excluding biologics here since that's not within the domain of our interest in the Center for Research on Complex Generics. The second and third questions were, which techniques and tools should we focus on? And the third question was, what training should we focus on? But with regard to this first question about which products to focus on, each participant was allowed to select up to two. So when you see some of the results, they add up to more than 100% because folks were able to select more than two. In fact, the vast majority of participants did select two. And the options here were complex injectable formulations and nanomaterials, complex mixtures and peptides, drug-device combination products, inhalation and nasal products, long-acting injectables and implants, ophthalmic products, and topical dermatologic products, as well as other. The second question, as I think I briefly mentioned, was which analytical techniques and tools to emphasize now. Again, excluding biologics, that's not within our domain in this collaborative agreement. And the options were data analytics and machine learning, local acting physiologic-based pharmacokinetic modeling, oral absorption models and bioequivalence, quantitative clinical pharmacology, and patient substitution of generic drugs, as well as the other category. And the third question dealt with educational topics. And folks were able to select up to 4. And the answers, the potential answers were really just a combination of what we saw in the first question with regard to products and what we saw in the second question with regard to techniques. So again, just kind of emphasizing that any one person was able to select up to 2 and 4 with regard to questions 1, 2, and 3 that concerned products, techniques, and educational topics. We also asked some demographic information. We want to know who was answering the questions, at least in terms of the demographics. One question was, were you willing to be a point of contact for your company? And much thanks to the almost 50 people that were willing to do that. And we also ended up doing analysis, as you might imagine, among some of these demographic questions about the answers that people provided. We also asked a employment perspective question of the respondents, and there was 13 options. For example, were they a generic drug industrial scientist or manufacturing personnel? Were they an executive or management person in generic industry? Were they, for example, scientists at a CRO, CMO, or CDMO, etc. We also asked people about the number of employees at their company. That ranged from 1, I think, all the way up to over 10,000 employees at their company. And then an additional demographic question concerned about their employer's interest, and there was 11 different options, and folks could select any that applied. For example, a lot of respondents worked for an API manufacturer. And they were also a finished dosage form manufacturer. And then, of course, there was other types of options to select from also. In terms of methods of this survey, again, there was 3 main areas of questioning that concerned which products to focus on now for the center, which analytical methods to focus on now for the center, and which educational topics to look at. The survey was approved by the University of Maryland IRB. It was open to the public at our website, complexgenerics.org, during essentially the months of December 2020 and January 2021. We emailed the information about the survey so people can go to the website to our growing email list. Again, I would encourage you to sign up if you have not already done so. Really much thanks to the many organizations that created awareness of the survey. I think in general we're actually quite happy with the survey response, in no small part because of several organizations. There was only one required question to the survey, and that was consent. If people didn't consent, then the survey was terminated. And with regard to the results, brief mentioning of some of these statistics, we perform stats in SAS, relying on the chi-square test largely. And since we had lots of questions, we corrected for multiple comparisons by Bonferroni correction. Okay, in terms of demographics who responded, we had 281 examined responses. That is, there was 281 respondents who provided consent and answered a question. Something that was a little bit surprising to me is we had quite a few people who gave consent and then didn't answer any questions. I guess there's a term in real estate called looky-loo, people who kind of want to look through the house but just there to maybe see, which was just fine. But we didn't count those people as being respondents. Over half of all respondents were, including so-called points of contact, were generic drug employees. So we think we hit things pretty well there, although we were broadly interested in hearing from the public. Respondents were equally distributed between being employees at companies with less than 1,000 employees, as well as over 1,000 employees, although there was a slight tendency for points of contact to be from larger companies. A majority of respondents were employed by a finished dosage form manufacturer, and about 1/4 were employed by API manufacturers, and in many cases they were one and the same. Again, we did do quite a few comparisons. For example, we compared the responses of employee points of contact as well as non-points of contact. I think we had almost 50 points of contact, individuals who are willing to represent their company. And just kind of looking at some of the results here, there was actually never an effect. So points of contact and non-points of contact gave pretty much the same answers. At least we couldn't tell the difference. We also looked at employer interest as being variable in terms of responses. And in the area of analytical methods, there was a difference between generic drug employees and non-generic drug employees. Maybe the thing that was most surprising to me in terms of demographic effects were company size. Where company size, for example, large versus small, had the biggest effect, and in fact it impacted results in all three question areas. And maybe many of you are perhaps not surprised by that. But yeah, we'll take a look at some of the results. So here's maybe the most informative slide of this presentation. Here are the top three replies from all respondents. Again, we have questions about which products to look at in terms of emphasizing for the center, analytical techniques to look at, and educational topics to focus on. And in terms of just rank order, first, second, and third. Before I start talking about what's in this table, I should probably say I don't want to overemphasize this either, because maybe this is a theme, is really there's quite a few product areas that were demonstrated to be interested, I'd say, across this survey. Nevertheless, I think we can say there was a rank order in terms of what was sort of on top. And for the question about complex products, here they are: complex injectables, formulations, and nanomaterials. Second place, drug device combination products. And third place, inhalation and nasal products. Educational topics, which was really the options were just pooled responses options from the first two questions, mimicked the responses for educational topics was really very similar to complex products. Same first entry here, top entry, same second top entry here in drug device combination products, and then coming in third here, which kind of matched the analytical techniques. Third place was data analytics, including quantitative methods and modeling and simulation. And with regards to the second question, analytical techniques, coming in first was locally acting physiologic-based pharmacokinetic modeling, oral absorption models, and bioequivalence in second, and then data analytics and machine learning in third. So I think this is going to have, some effect as far as what priorities are. It doesn't quite answer the question, what specifically should we be doing. So I will talk a little bit more about that, and hopefully I hope to learn more today about that level of detail. Okay, before we sort of dive into some of the tables, let's take a look at some of the findings. For complex products, points of contact and non-points of contact didn't give any perceived differences. Employer interest was also not a factor, whether what the employer's activities were. For example, generic drug employees didn't differ from non-generic drug employees. Companies with more than 10,000 employees did differ from all other companies in terms of inhalation and nasal products, where they emphasized that more compared to the smaller companies, I guess. And there also was a difference between large and small companies. What I mean by large is over 1,000 and small, under 1,000. With regard to larger companies, did emphasize complex injectables, formulations, and nanomaterials. With regard to, in terms of additional summary findings for analytical methodology, that second question, again, points of contact and non-points of contact didn't differ. Generic drug scientists did differ from all others in terms of emphasizing locally acting, physiologic-based pharmacokinetic modeling. Generic drug executives did differ from others in terms of the other option. I think maybe a quarter said, there's other analytical methods that need prioritization. So again, just going back a slide or two to look at the top three, I'm not sure that tells the entire story at all, actually. Although, having said that, it's nice to have some prioritization. Also, in terms of analytical methods, large company versus small company did differ with regard to locally acting PBPK modeling, with large companies emphasizing this as a relative priority. With regard to the third question, educational topics, points of contact and non-points of contact did not differ again. They never did for any question. Employment interest was not a factor. Didn't matter whether you were a generic drug employee or not. Gave the same answers as far as we could tell. There were some differences with regard to company size. Companies with more than 10,000 employees did differ from all others with regard to locally acting physiologic-based pharmacokinetic modeling, as well as data analytics. There was a fourth question, harmonization, which was, we'll take a look at those results, very, very highly supported. So overall, definitely would have to say that these results will help prioritize the center's research and training efforts. Okay, let's take a look at some of the results in tabular form. So think with regard to the first question about complex products, again, individuals were able to allow to select up to 2, and the vast majority did do that. Red here indicates what came in the top 3. Again, complex injectables, formulations, and nanomaterials, drug device combination products, and inhalation nasal products. But again, I would have to say many of these other areas did receive substantial voting, shall we say. I'm not an expert on ophthalmic products, but which is a somewhat narrow area, I would have to say, but still 13% actually did tally for that. And then taking a look at complex products by number of employees, again, larger companies, those with more than 1,000 did have a preference to emphasize complex injectables, formulations, and nanomaterials, as well as inhalation and nasal products. With regard to the second question, analytical techniques, again, in red here is showing and these are shown in terms of order in which they appeared in the survey, as in the last slide also. These are the top 3 that we talked about. But again, I think maybe I would just emphasize that, just because it's not in the top 3 doesn't mean it didn't receive some substantial support. Here's a topic I happen to be personally interested in, patient substitution in generic drugs, with a quarter of respondents selecting that for at least one of their two tallies. Looking at this question about analytical techniques by employer type, showing some of the larger employer types here in terms of who responded. Generic drug employees did put an emphasis on locally acting physiologic-based pharmacokinetic modeling, and generic drug executives did put some greater emphasis on the other category, seeing a need for techniques that are not reflected here. Continuing with our discussion about analytical techniques based on number of employees, so larger companies, those above 1,000, and in particular those with more than 10,000, gave a little greater emphasis for locally acting physiologic-based pharmacokinetic modeling. Going to the education topic, in red here are the top 3 that we previously discussed. I guess the other thing that would just sort of jump out at me would be a number of areas actually did receive really quite a bit of support otherwise also. So clearly there's lots of needs in this complex generic space. And just showing some of the demographic information about educational topics, in particular number of employees, maybe nothing here. Maybe look at this later at your leisure. In red here we see data analytics as well as locally acting physiologic-based pharmacokinetic modeling being more emphasized by the largest of companies. There was just very, very high support for harmonized approach when it comes to complex generics requirements. You can see the numbers here. And I guess my 2 cents would be this is sort of something that's probably hard to ignore. I think I understand that individual agencies, for example, the likes of the FDA, do need to worry about their sort of home country, maybe even prioritize that. But having said that, these numbers are really very, very supportive of always keeping this harmonized international approach maybe in mind. So that was a brief tour of the survey. Really super thanks to all those who participated. If there's a need, if you think there's a need to maybe do this again in maybe 10 years, 2 years, please let me know. Again, much thanks to the several organizations that create awareness of this survey, since it is, I think, going to be informative. Again, getting back to maybe a generalized question today about what research is needed. Again, hopefully you found this survey very helpful, but I guess I would also quickly point out of the limitations of the survey. It probably doesn't give us the granularity in terms of maybe things that we can decide to do. And in fact, as Anna will describe, we did do some reach out to companies, have some informational sessions, which definitely were provided a lot of helpful insight, and hope to learn more exactly about what specifically merits attention today. Again, please contact us. Again, my name is James Polli, and I will be speaking next. We're the co-directors. Also, a very important person is Vishal. She's the manager for the center. Please email us, and also please visit our website and sign up for the listserv. Thank you very much.
In this presentation, I am very happy to share with you summary of interview feedback from industry stakeholders. As you know, communication is a central mission of CRCG. Through this communication, we hope to establish a neutral channel to facilitate interactions between industry and the FDA. Use this channel to outreach to industry to understand what are the main pain points in development and approval of complex generic products. We also hope to use this channel to solicit feedback from the industry stakeholders on product-specific guidances and other guidances that had been developed by the agencies. We plan to publish our research and peer review publications and use to assess the metrics of CRCG success. Prior to the interview, we sent an email asking several fairly open-ended questions. We wanted to understand what are the main barriers for development of complex generics and what are the industry pain points around those products. Are those barriers due to scientific issues, regulatory challenges, market competition, or ROI considerations? We also asked about how the center could help the development of complex generics through either research, training, or facilitating communication with the agency. Some interviews were very well structured and the company came prepared with slides and written summaries, and some were not, and all of the interviews allowed for an open dialogue. This information will be used to focus the efforts of the center as well as to help FDA's ongoing GDUFA funded activities. We plan to publish the outcomes of this interview in a peer-reviewed publication. In total, we interviewed 20 organizations, including some of those interviews were conducted multiple times. The type of products discussed included complex injectables, liposome, long-acting injectables, iron colloids, inhalation, topical and transdermal products, ophthalmic emulsions and suspensions, and orals. The main issues brought up by the companies included CMC issues for either specific products or product classes, old and variable products, bioequivalence issues, clinical trial design, use of modeling and simulation, product-specific guidances and the changes of product-specific guidances, general communication concerns, leachables and extractables, and nitrosamines. Here we summarize the companies we reach out to. Those include large multinational generic companies like Perrigo, Apotek, Teva, Viatris, Sandoz, Cipla, Amneal, and Sun Pharma, as well as the smaller specialty companies such as Fresenius Kabi, Cassette, Solaris Pharma, Nexus, Xillia, Capstone, as well as trade organizations. We also highlight the type of products that have been discussed during the interviews. I would like to use this opportunity to let you all know that if you are not on this list, please reach out to the center and we will be happy to talk to you, as well as the fact that we would like to continue those interactions on an ongoing basis and perhaps interview you again in the next 6 months as the field is constantly changing. As you can see by this table, the number one concern brought up through those interviews were specific issues with chemistry, manufacturing, and controls. Those issues were related to either specific products or broadly to general product categories. They also included drug-device combinations, as well as important issues around old and highly variable products. Companies also discussed specific bioequivalence and clinical study design issues, as well as changes in product-specific guidances, leachable and extractables, and nitrosamines. The first item I would like to discuss is the use of modeling and simulation to supplement bioequivalence studies. It is absolutely clear that modeling could help development of complex generic products. The number one product category brought up when modeling was discussed in those interviews were utilization of PK/PD modeling for inhalation products. It's also as well as for oral dosage forms. For products like ophthalmic, topical, and oral inhalations, the in silico modeling is very helpful. It's also generally agreed upon that populational pharmacokinetics to discuss the differences of the site of absorption is very useful, and modeling around long-acting injectable studies also could be beneficial. However, industry had a number of major concerns. They all understand that it's important finding alternatives for performing of expensive clinical endpoint studies, but no single company would like to be a trailblazer in this area. There are also kind of lack of general models that are accepted by the industry and the agency for complex generics. What we realize that GDUFA research sponsored development of multiple models, but also the companies themselves either use software or have their own software written to predict modeling of deposition of inhalation products, for example, and the impact on pharmacokinetics and bioequivalence. But there is not a single model that is acceptable for everybody and agreed upon by the agencies. They also think there is kind of general lack of publications on what are the expectations for the bioequivalence modeling and what would be required to validate the model. Everybody believes that they could really benefit from a FDA feedback during model development, and companies would really like to have meetings to discuss their approaches as they develop their models. While modeling is often used, nobody knows how modeling will be reviewed by the agency and whether some of those modeling approaches will be accepted. And this increases regulatory uncertainty around the use of modeling for generic products. There was very specific questions. For example, for cytotoxic drugs, many times some of the time points are missing, and companies were wondering if they could use modeling or partial modeling to supplement some of this patient data. About 60% of our time during the interviews were devoted for different chemistry and manufacturing control concerns. The number one concern is development and validation and the lack of standard analytical methodologies for physicochemical characterization of complex generics. I think as an industry, we're all aware how to establish and validate a typical HPLC method. However, anytime we're talking about characterization of complex products, there are no standard methodologies out there. For example, it's not clear how to measure particle size and morphology, how to conduct in vitro drug release testing, how to assess peptide immunogenicity, patch adhesiveness. There are also no clear guidelines of how to validate this method. Some of those methods are very difficult to validate. It's also not clear how best to show their discriminative ability. Many of those methods really are highly variable and therefore cannot be held to the same standard as a typical HPLC type of assays. There are another issues around analytics that was brought up by several companies that they develop more novel, discriminative, or highly sensitive methodologies that well accepted within the industry, but when they submit those results with filings, often they are not accepted by the agency because they do not match USP methodology that many times not as sensitive or not as accurate. There are also a number of so-called boutique methodologies that are brought up by the agency in their publications, and the companies don't have access to this fancy equipment and do not know if it's expected for them to use those methods to characterize their products. Those are morphologically directed Raman spectroscopy, various imaging techniques, and small angle X-ray diffraction methods. The next issue brought up was it's a very extensive analytical requirements discussed in product-specific guidances. Several companies said it pretty much this way. They said there are so many parameters listed as important in the guidance. Are all of them equally important? Do we have to measure just some of them, or are we required to measure all of them? And they have their thinking that many of those parameters really do not impact by equivalence, and yet they have to measure and match them. Some of those requirements appear to be almost of purely academic interest, and there are no specific scientific rationale or data that supports that this particular requirement is important for product bioequivalency. Some of the examples were hydration layer for the liposomes and particle drug distribution within the pores of long-acting injectable microsphere products. Other general CMC issues included, do you have to be Q1, Q2, or can you be Q1, Q2-like? In many instances, company believes that being not precisely Q1, Q2 within 5% of variability still results in products that are bioequivalent. And in many instances, there are some patent considerations that box the companies out from achieving the complete Q1, Q2. But there are no alternatives of how to use if Q1, Q2 is not fully achieved. In many instances, product-specific guidances are so detailed that Q1, Q2 route appears to be almost more complex than clinical endpoint studies. It's also difficult to achieve Q1, Q2 equivalence for products that have over 10 ingredients. Specifically, companies brought up many topical products, including petrolatum and waxes, and difficulty in figuring out exactly what excipients are there and matching Q1, Q2 for the specific excipients that are only 1% in the formulation. In addition, companies mentioned there are some specific, very small issues in Q1, Q2, like a buffer composition or osmolality that holding off product approval. In addition, there are some products where clearly there is a difference between the label provided by innovator company and the data reverse engineering that was achieved by the company. Other general CMC issues included RLD, API, and excipients availability. It is difficult to procure 3 lots for some of the drugs, especially drugs with low volume, orphan drugs, or drugs that are very highly priced. There is a requirement that you have to have N equals 3 of everything, and for some of those RLDs, only 1 batch is produced per year. Some of the excipients, only 1 batch is produced per year, which means that the company has to collect batches for 3 years before the start of reverse engineering and bioequivalence. And some RLDs are just no longer available. The next issue, very important, is around highly variable or old RLD products. There are several products, and they appear to be very important products with high markets and no available generics. They themselves are highly variable. If you buy 2 lots of the RLD, many times they wouldn't pass bioequivalency or in vitro comparability when compared to itself, based on drug release rate, particle size, or purity. When bioequivalency studies are conducted using 2 different lots of the products, again, many times variability is greater than the specifications set by the agency on bioequivalence. The products that were mentioned in this respect were iron sucrose, PLGA, long-acting injectables, dose-metered inhalers, and topical steroids. Additionally, there are clearly some discrepancies between label and actual products, such as drug overages, which make it very difficult to develop generic products and file. For some of the products, there are lack of availability of API and excipients, especially for some of the old products, and again, some of those batches made only once per year, making it difficult to collect 3. Additionally, some of the old products, and in a way easy to manufacture, they produce using very old technologies. Examples were given like estradiol creams and other topical steroids that could, the particle size lot to lot is so variable. The company would like to use more novel methodology to control the particle size, but then they do not match the variability of the initial product. In some instances, the formulations are not very stable, and the company would like to add a small amount of antioxidants to stabilize the formulation, but then the formulation will not match Q1, Q2 requirements. This slide highlights some of the most frequently discussed products during the interviews. Liposomal products, long-acting injectables, and iron sucrose and ophthalmic emulsions were brought up by a number of companies. For liposomal products, there is difficulty in development and validation of some standard analytical methods to characterize particle size, particle morphology, in vitro drug release, lipid impurities. There are also many parameters required to be measured by product-specific guidances, and many times the company already completed bioequivalence study and they have to go back and measure many parameters. There are also sometimes products are morphologically different, but they behave identically in vivo. For long-acting injectable products such as PLGA, similarly many methods were highlighted, such as morphology, porosity, polymer purity and particle size, in vitro drug release, and also drug distribution within the microspheres. Characterization of peptide aggregates within the microspheres and potential impurities impacting immunogenicity in complete drug release were mentioned by several companies, as well as RLD variability. Iron sucrose was a big topic with difficulty in methods and drug variability, and also ophthalmic emulsions. The same, difficult in characterization, variability of RLD, and also it appeared to be some small Q1, Q2 issues preventing approvals. Inhalation products are very unique. It is like a combination of the entire pharmaceutical science field in one type of drug. They are also very important product for the industry, and many companies are working on dose-metered inhalers because those are big products and there are very few generic versions available. Yet they represent very unique challenges. especially for poorly soluble in drugs and dose-metered inhalers. There appears to be substantial batch-to-batch variability in the innovator products between particle size and morphology. The product also requires drug-device combination, and therefore there are certain patent considerations around the design of device and how the patient end up using generic device versus innovator device. There are clearly big differences in how patients use the device and populational difference in pharmacokinetics and response. Those type of products require very large clinical studies. People estimating it's about $30 million studies to show clinical bioequivalence. And therefore, companies really would like to have the ability to discuss with the agency prior to the start of the studies. Another issue with the entire development and clinical trials and development of dose-metered inhalers that there appear to be difference how they're regulated in European Union versus in United States. Those products require development of standardized method and standardized models. Methods for defining particle size morphology, development of physiologically relevant dissolution methodology, as well as extensive modeling needs around pharmacokinetics, PBPK, as well as populational pharmacokinetics. Topical products were brought up by a number of companies. In general, industry is very appreciative of new guidance on IVPT studies that could be used instead of bioequivalence studies. However, there are a lot of outstanding questions around the design and conduct of those IVPT studies, and some of the statistical considerations that are not covered by the current guidance, and how to deal with the outliers, what to do, how to assess the impact of crystal formation, how to assess the impact of permeation enhancers. It's difficult to meet some of the specifications due to high variability, difficult to find n equals 3 of excipients and lots, and difficult to analyze the products that have many ingredients, over 10 ingredients. It's very difficult to establish Q1, Q2, but some of the excipients are only at 1%. The next couple of slides are focused on the clinical trial design issues. Several companies brought up the design for large endpoint trials for inhalation products, and their strong desire to look into alternatives for those endpoint studies based on PBPK modeling and populational PK modeling. Around the standard trial design and duration for bioequivalence studies, companies discussed long-acting injectables just because those trials are very lengthy, hard to recruit patients, and they would like to use alternative modeling-based approaches to shorten the duration of those trials or reduction the number of patients. They also want to look into the possible alternative for bioequivalence studies based on IVIVC or in vitro characterization. There is a limited number of patient pool, especially drug-naive patients for cytotoxic drugs. Another category discussed was poorly active topical products that are not Q1, Q2 equivalent, and how those studies large in variables and many times more expensive to prove equivalency of generics than approve the RLD. There is a very strong desire within generic industry to strive toward harmonization of clinical trial requirements between FDA and EMA and other regulatory bodies. This could really reduce the cost of clinical development of the products. There are issues around RLD sourcing for low-volume drugs, orphan drugs, or no longer marketed products. There is the need to source products specific for geography, such as Europe versus US, and in many instances the product is exactly the same and produced in the same plant. For some trials, the products expire before the clinical trial completes. There are other bioanalytical concerns around how to measure free versus encapsulated drug and plasma for liposomal products. or how you measure free or bound iron for iron colloids. And companies have examples of multiple bioequivalence studies failed due to product variability or the lack of robust analytical methods. For LAR products, discussions were around site of injection versus bioequivalence, and as well as correlation between adhesion and bioequivalence for transdermal products. Leachable and extractables is an important issue that generic industry faces that came up in a number of interviews. The main issue is the lack of consistency in how applications are reviewed. One company gave us an example of the same product submitted either by the manufacturer or by the distributor, absolutely identical end application, and one comes back with no deficiency and another has pages and pages of leachable and extractable deficiencies. Those inconsistency of the review in leachable and extractable cause a lot of delays in product approvals. Many companies see 9 out of 10 programs, so 80% of ANDA delays due to leachable and extractables issues. There is a need to repeat expensive studies, decreasing commercial viability of the products. And even low-risk products had to go back to generate additional leachable and extractable data. The company is sometimes unclear on the FDA expectations, and they would like to have an opportunity to discuss with the agencies their data and their deficiencies. They also see the same number of scrutiny applied, for example, for the cream for container, whereas the cream is stored for over a year, and for the applicator that sees the cream only for 1 or 2 minutes. This slide summarizes general communication and guidance concerns. Everybody loves pre-ANDA meetings. Companies find them very useful, and they would like to have another useful meeting in mid-review or post-CRLT where they could be able to engage in dialogue with the agency to defend their position or their application and to actually receive detailed feedback through this communication. In general, there is a desire for improved communication and feedback on expensive trial design or design of human factor studies or leachable and extractable studies. Another concern is companies would like to receive all the deficiencies during the first review cycle. Many mentioned that they submit their answers to the deficiencies only to receive new set of deficiencies during the second review that were not previously mentioned. Missed GDUFA goal dates due to complex review issues also appear to be concern for select products. Companies love product-specific guidances. However, they have the strong desire to be involved in product-specific guidance development, or at least understand where FDA coming from in the development of the guidance and provide the feedback. The changes in product-specific guidance appear to be a major problem for the industry. Many times requirements are updated and the company have to go back to redevelop the product or repeat the clinical study, which really hinders economical viability of select products. It's also unclear to what standards the application already held to that had been filed, and also changes in the guidance means that later products held to higher standards than earlier generics. How the center could help the industry addressing those concerns? We could perform reverse engineering for some of those highly variable products that are of high interest to the industry. We could establish and characterize performance of non-standard analytical methods, including establishing boutique characterization methods and making them available. We can understand the impact of Q1, Q2-like or morphological different products on bioequivalence, develop libraries of RLDs, APIs and critical excipients and make them available to the industry, as well as work with the agency on development of analytical methods for upcoming new product classes, such as RNA therapeutic. How can we address modeling and clinical trial concerns? I think it's important to work together with industry stakeholders and the agency to develop so-called standard models for inhalation, ophthalmic, long-acting injectables and topical products. We could also jointly develop an understanding of what industry requires for model validation. In collaboration with industry, we also would like to explore alternatives to bioequivalence studies based on in vitro characterization and IVIVC, again for select high-profile products, and re-examine bioequivalence requirements for those highly variable RLDs. We also would like to attempt to work with FDA and other regulatory agencies on harmonization of clinical trial requirements. Additional concerns including working with the agencies to address leachable and extractable review issues, which appear to be an important problem concerning both new drugs and generic products, as well as develop analytical characterization methods and risk assessment for different nitrosamines, organize workshops on specific complex products or specific newly developed regulatory guidances to both allow for the agency to provide clarifications and also to allow for the industry to provide the feedback on how those guidances affect them. Also, we hope to be able to foresee potential scientific questions for products with patent expiries in the next 5 to 10 years, as industry is already working on those products. This brings me to a final acknowledgement slide, and first of all, I would like to thank the agency for awarding to us this U18 grant from GDUFA funding. I also would like to acknowledge multiple generic companies that agreed to be interviewed by the center. We're really grateful to Dr. Sam Rainey, the FDA grant manager for this grant. Sam not only helped us to set up those interviews, he participated in many of them, and he helped us to shape the vision for the center. Dr. Lisa Park from AAM helped to facilitate some of the interviews. I will also like to acknowledge Dr. Jim Polli from University of Maryland, the center co-director, Dr. Visha Krishnan, the center manager, and my graduate student, Jill Coughlin.